The dual DNA rule assay can provide mechanistic insights for ribosome translocation and probe other nucleic acid displacements. Our dual DNA rule assay is currently the only method that can objectively probe both the entrance and exit sites of mRNA in the ribosome complexes with single nucleotide resolution.
Similar methods are also applied to measure DNA binding strengths and the selectivity of chemotherapeutic drugs. Visual demonstration of the method is a good chance for us to intuitively show the advantages of our technique and help other researchers to further develop into more applications.
To begin, make a plastic sample well with a styrene strip by drilling a cube in the center with dimensions of 4 mm by 3 mm by 2 mm. Then, using epoxy, glue a piece of biotin-coated glass of approximately 5 mm long to the bottom surface. Add 20 microliters of 0.25 mg per milliliter streptavidin aqueous solution into the sample well and incubate at room temperature for 40 minutes. Then rinse the sample well twice with TAM10 buffer. To immobilize the ribosome complexes without antibiotics, first remove the buffer from the sample well and add 20 microliters of 0.1 micromolar MF pre or MF post ribosome complex into the sample well. Incubate at 37 degrees Celsius for 1 hour, and then rinse once with TAM10 buffer. The ribosome complex now binds with the streptavidin on the surface via the 5' end biotin on the mRNA.